Good morning, everyone. Welcome back to day 2 of this workshop. Just a reminder that we talked yesterday about cancer-specific considerations and computational approaches in relation to multi-omics studies. We heard about many challenges, but we also had some really nice discussion about what methods, approaches, study designs, and exciting new research is attempting to move both of those aspects forward. We're going to cover 2 additional sessions today and then do a final wrap-up session this afternoon where we try to pull it all together. So we are encouraging everyone who can stay on for that final session and help us with the final recommendations going forward to NIH from this workshop. This next session today will be addressing environmental exposure specific considerations. So we're going to be discussing a lot of the challenges specifically with integrating a lot of different environmental exposure data with other omics data. And so I would like to introduce Dr. Gary Miller, who's from Columbia University, to help to moderate this session.
Okay, thank you, Kim. Yeah, it's my distinct pleasure to help moderate this session. We have a distinguished panel of speakers and panelists today. First up is Dr. Dean Jones from Emory University, who I've worked with for many years. And I think that when we think about the integration of these different types of data, that Dean is a key person to blame for having so much of this data. He's been working for many years to collect as much information as we can about our exposures in biochemistry. And it's led us to this point where we're now struggling with all of this. I also think his work in different animal models and biochemical models has helped kind of deal with how you integrate this into a better understanding of the biology. So I think it's a great framework for this. So with that, I will turn it over to Dr. Dean Jones.
Okay, thank you, Gary. Let me see if I can get this moving now. And hopefully that.
Perfect.
Disclosure, and that is that Gary always describes me as a crusty old man, and I'm absolutely not a crusty old man. I'm a barnacle, and I'm just clinging to the side of a whale that is environmental health and enjoying the ride. So with that, I'm going to go ahead and try to zoom through some slides here. I'm going to talk first about what Gary alluded to, and that is this metabolomics and exposomics collision course. I started several years ago, actually 2 decades ago, trying to take metabolomics to an omics scale. And for this, I followed the principles that Elias Zerhouni, the former NIH director, outlined for the data of the future, and that's these 6 characteristics. And with that, over the years, and with a lot of talented and very hardworking colleagues, managed to really push this forward to the point where after about 15 years, we were able to measure up to 1 million chemical signals in small biological samples. And so we call this high resolution metabolomics. It really has sort of morphed into exposomics because that's really what it is. I have outlined this multiple times in the past as far as what is captured in this high resolution metabolomics that really is representative of the exposome. So what I have to point out, and I think everyone is fully aware of, is that environmental epidemiology generally works with a relatively small number of targeted measures. It really is a Cartesian reductionism approach. The reality is that today, exposome epidemiology is possible. That is, examining all of those exposures at an omics scale. And so Barbara Cohen and I had an article just a few years ago on really providing a vision for exposome epidemiology. And the way we defined this was really using omics scale biomonitoring. And so that's outlined here. And I'm not going to go back. I'm not going to spend a lot of time on this because it's been presented in multiple publications. Lots of needs, lots of details as far as development of exposome epidemiology, but I think that's really what we have to do. So as we're thinking about the integration of all these omics layers, we have to consider the exposome as one of those omic layers. And the simple principles, simple rules are, we don't live with one exposure at a time. Lifelong exposures are cumulative. And so what I believe is that the omics scale analyses give us an opportunity to explore the multiplicity of exposures and the complexities of their interactions, especially with the biological system. So quick snapshot of where we are. Multiple labs now have the high resolution mass spectrometry coupled to gas chromatography with capabilities for measuring in the range of 1,000 known environmental chemicals in small biological samples. In principle, with an untargeted approach, we can get over 100,000 chemical signals from these analyses. For liquid chromatography, it's even more powerful because there's multiple platforms that are available. And we have Tom Metz on the panel, and he's really an authority in this area. But the reality is we're now able to capture in the range of 1 million chemical signals. Now, it's not that many chemicals, and individuals aren't going to have nearly that many chemicals in them. But collectively over the populations and different geographies, we have to anticipate that there are 1 million chemicals that we're exposed to. So we heard yesterday about the FAIR principles for data management in this field. I think what we have to do is combine that. It overlaps with Eliza Zerhouni's principles, but we have to combine that and come up with a set of 8 principles. Won't go through these, but they're all there, and I believe they're obvious, but we just need to pay attention to them and really try to obey them the best we can as we push forward with the human exposome research. Now, second point I want to get to is the concept of multi-omics integration. So this is really the whole purpose of this session this morning. And I have to cover it from 2 directions. One is the issue of integration of omics data in human studies, but also then the translation of that information to model systems. And as Gary already indicated, I've done a lot of work on studying doing mechanistic toxicology research in environmental health. And so this is quite relevant. So from the model systems, central dogma really tells us how to think about this in the omics world. And that is, we have omics capabilities for the different layers within central dogma. And we can bring those into alignment or integrate those with other types of data, such as immunological measures, microbiome measures, and even exposome measures. And so that's what's critical in terms of, as we're moving forward, we want to be able to do this in a mechanistic way and then be able to capture the breadth of our exposures. So software that Karan Upal wrote when he was here with us at Emory, it's called XMWAS, and this is what we use most typically. It's a data-driven integration. It's very logical and it's straightforward so that really untrained individuals can come in with omics data and begin to explore the community structures and the network structures, and then be able to go take that information back to the resources, the abundant resources that are available to begin to get biological understanding. And so what this does is we start with pairwise correlations of whatever the arrays are, and you can put in multiple arrays. It's fairly convenient to do 4 different omics layers together. and combine that with other types of non-omic data, and can do pairwise correlations and analysis, and then basically do correlation-based ranking and then filtering based upon that. So you can go at either a very, very dense structure, or you can go to a more sparse structure just by selecting your cutoffs for your correlation. Similarly, statistical, you can perform different types of statistics, and you can do statistical filtering to be able to further refine what you're looking at, and then use community detection tools such as some that were discussed yesterday, and visualization. And I will comment that visualization may be a point that we need to specifically pay attention to, because with these complex data, we need to have some guidelines and some common ways to make it easy for everybody to communicate for visualization of these complex systems. So the added value of these community detection tools, which I don't know that has been really emphasized, is that you can get quantification of the centrality of different components within this complex network structure. And then you can look at differences in centrality between two conditions or two variations. So in this case, these are the delta centrality measures of specific cytokines and chemokines before and after influenza viral infection in mice. And so it's quite clear that you can see the community structures change, and you can see how central different components are, and how much of a change there is. So we've applied this in many different cell and rodent models. And specifically what we do is we vary some exposure, and then do integrative omics. And these are the 5 integrative omics that we have worked on mainly in the model systems. And then we can use these for measuring functional responses. And as an example, and there are many of these in the literature, so I don't need to, and I'm sure most are familiar with these. It's straightforward. In this experiment, we varied manganese dose in a cell culture study, measured mitochondrial activity, and then measured other omic measures. We did, in this case, redox proteomics, metabolomics, and transcriptomics. And then we can take that data in a network analysis and specifically look at the central hub of physiological response to manganese. And this is right here, I highlighted beta-hydroxybutyric acid, one of the very important intermediary metabolites. And you can differentiate between physiological networks and toxicological networks. So once you increase manganese to a high dose, it becomes a toxicologic exposure. And then you can look at, you can expand the view and you can zoom in, you can look at specific metabolites that are in this central cluster. You can look at correlated proteins. In this case, these are redox proteomic changes, proteins that are being changed in oxidation state. And then you can look at correlated transcripts. And then, of course, these type of data can be mapped out using online tools to functional pathways. The value of these model systems really is that you can test those network structures. And you can do this through genetic manipulations, through pharmacologic manipulations. You can use flux analysis, so you can actually determine whether there's a change in the rate of that beta-hydroxybutyric acid metabolism. And so you can do other mechanistic tests. And so this is the real value. Now, as transitioning to human studies, what I wanted to point out was that these model systems are also valuable because they can allow us to discover molecules that were chemicals that we didn't know were even there or didn't know what their functions were. And this is one that we described a couple of years ago. It's delta valerobetaine. This is an interesting one because it is produced by microbes in our gut from dietary components, specifically lysine and lysine-related metabolites. And when it's converted to this valerobetaine by the microbiome, it becomes a diet-dependent obesogen. And this is purely discovery. This was not, at the time we did this, this chemical was not even known. And so it's extremely valuable. And in a translational sense, we can then take that chemical and go back and look in human populations. And indeed, when we did that, we found that valerobetaine is associated with adiposity in humans. And also others have shown now that this is associated with fatty liver in humans. So it's very important to use these, I think, these untargeted, very powerful untargeted methods to really capture and be able to integrate the data. So this is an example which provides a different take on all of these omics integrations. And this was a study done with Sushma Cribbs, who is a clinical scientist who studies individuals with chronic HIV-1 infection. And so she had collected bronchoalveolar lavage fluid from individuals, and we took advantage of that by measuring the microbiome and the metabolome in those samples. And what was most remarkable is that when we did the integration, what we found was that the metabolome microbiome interaction in these otherwise healthy HIV-1 infected individuals was actually centered on 4 groups of microbes that are the leading causes of opportunistic infections in this population. And this is done without any knowledge of what the chemicals are in the metabolome. So this was a totally non-targeted analysis. And what it shows is that the central organizational structure, the biological network structure, is identifiable regardless of identification of all of the elements of it. So quickly now, I've obviously used more time than I had thought I was going to use, but I'll go through this quickly. I want to illustrate this one study that was published last year. Doug Walker, who's one of the panelists, is a co-author on this, as is Gary Miller. It's a project led by Kostas Lazaridis at Mayo Clinic, and it's on primary sclerosing cholangitis. This initial publication it was hundreds of individuals, but it's actually a pilot study because honestly, we had no way to power the study. So without any prior study of its type, we had to be considered a pilot study. But Doug did this extensive EWAS, MWAS integration. I don't have time to go through the details. It's published. And the main point is that using an untargeted exposome approach, he was able to pick out many chemicals that are actually associated with the disease. And then when he did an integration of these environmental chemical measurements with the metabolomics data in same individuals, what he found was this exposome metabolome interaction network, which included preservatives that we eat, fungicides and insecticides that are used commercially, as well as unfortunately a surfactant. And I say unfortunately because for the past 3 years with COVID, we have all been disinfecting ourselves with these surfactants without really paying attention to what the burden is that we have in our bodies. But anyway, these can be mapped to pathways. And so even though it's purely a correlative study, it provides a lot of potentially mechanistic information and insight into disease processes. I wanted to mention this study by Shizao Li, also done when he was at Emory. Shizao is a very important data scientist and has really made important contributions in terms of how to deal with this untargeted data. And he described this hierarchical community network approach. And the most important thing about this is it starts off knowledge-based. So in other words, there's knowledge-driven data reduction prior to the data integration. And it's a very nice example that's worth reading as far as if you're thinking about getting into this type of analysis. The main reason I mentioned the hierarchical, it's a fabulous paper, but the hierarchical concept is one that we need to pay attention to because it's very possible that any of the integrative omics may be important in terms of describing biological networks. Critically, if these are overlapping from different studies, different populations, different diseases, if these are overlapping in their structures, or if they're present with common hierarchies, then if we create a cancer exposome atlas, then the network structures may emerge naturally just from the cumulative data. And this is why I believe it's not a question of can we do it, it's a question of we must do it as far as development of a cancer exposome atlas. So I'm going to finish up briefly with this concept of dose versus burden, which I had in the title. And the real issue here is that when we're dealing with most environmental measurements that are external to the body, we can talk about the dose of exposure, but we don't really know what the cell or tissue burden is for that exposure. And I mean, everyone's aware of this, but the question is it a translational pitfall? And I have to say, of course not. It's just a gap that we need to fill. And so we've started efforts, again, pilot studies of how to do this. And one of the ways that I think is going to be very powerful is this exposome-metabolome interaction structure that I've already talked about in terms of the human studies. And so in this case, we used, this was a study with Colin Weber, Joe Sharma, and some of the surgical team here at Emory. They remove portions of the parathyroid when there's for hyperparathyroidism. And so they collected a series of these samples, and we analyzed them by gas chromatography to get a broad number of environmental chemicals. And then we mapped those in terms of metabolic responses to which metabolic pathways are actually linked. And what was very interesting in this is that these pathways included energy metabolism and nucleotide metabolism, which are really essential for the growth. Now, I will say that this is not a malignancy. This is a neoplasm, but it's one that does have aberrant growth. So I think it's a very powerful approach. We've also done this with a metalome metabolome interaction structure. This is with cadmium. Cadmium is a carcinogen. Unfortunately, it's high in our fresh green vegetables that we eat in salads. And so we eat some of it every day if we have a fresh salad. And what we found is that in this human study is that the cadmium associated phenotype in human lung samples recapitulates what we find in a mouse model where we've dosed the mice with cadmium. And more importantly, is that in the mouse study, there is a cadmium selenium antagonism where the essential nutrient selenium protects against cadmium toxicity. I put some maps in here just to show you that, hey, there's quite a difference in the distribution of selenium and cadmium in the soils. And what's important is that we can map these to pathways. We can see that with the positive and negative associations with this central cluster of metabolites, that indeed the characteristics are that of antagonism. So again, I think there is a gap that can be filled by looking at the human tissues. That included normal and diseased human lung in that case. Another example of this type of an integration is a study that we did with Victor Darla-Usmar's group over at University of Alabama in Birmingham, looking at mitochondrial function These are in human platelets. So these are just collected from healthy people and measuring using the seahorse technology to measure mitochondrial function in the platelets, and then map that to the metabolites in the serum plasma. And the most interesting thing is that the primary variables in this untargeted high-resolution metabolomics analysis, the central components that were correlating with mitochondrial activity, were environmental chemicals and dietary chemicals. And finally, the example I want to show was one that emphasizes the geography issue that was apparent in that cadmium and selenium maps. But this is a geography lesson, and this is done with Arjun Arthria and Caroline Grant at Mayo, and it's also involved in that PSC cohort or PSC study. And in this case, it's a comparison of bile collected from patients in Norway and the US. And Caroline has done a wonderful study looking at the environmental chemicals, quite different in terms of US versus Norwegian population. The metabolome-wide association, quite different in terms of those structures. And then she's integrated those. And what the integration shows is that the network architectures are strikingly different. There are some characteristics that are similar. Populations are small here. These are small numbers, so we don't know how representative this is, even of the Norwegian and US populations. But the main point is geography is important, and geography is important in an integrative way, and we really have to map that out. So to summarize, I think where we are is that the omics scale, metabolome and exposome measures, need to be more extensively used in environmental epidemiology. And this is specifically relevant to the cancer burdens. The second point is that the integrative omics can be, incredibly insightful. And this is useful in humans because it leads us, it directs us towards mechanisms. And the mechanisms and the data provide us a common way to do the mechanistic studies in animals to actually replicate and test what we find in the human studies. And then finally, the gap between the mechanistic research and the epidemiologic research, that dose versus burden issue. I think we have an opportunity to really fill that gap, and that's specifically through using normal and diseased human samples, and just do this in a non-selective way. That was one of the aspects that was discussed yesterday extensively. We need to be thinking about this in a non-case control format, because we really need to be able to generalize it. And I think that's it. I ran a little over on time, but hopefully there's time still for some discussions, and hopefully I entertained you a little bit, and just remember that concept of being a barnacle on the side of a whale. It's how I feel, and just enjoy science.
Thank you, Dean. Yeah, we have time for a question or two. I'm going to kind of synthesize some of them, but one of the questions came up like with the inherent complexity of the mass spec metabolomic type data, and the question of, can that complexity ever become actionable, versus the more static aspect of the genome in that respect? Like, what are your thoughts on the actionability of what we can get out of this sort of analysis?
Okay, that's a great question. I have a series of slides on that because I believe it is actionable. If from a metabolic sense, if we can identify, we're getting a lot of mapping back to the linoleate pathway. Linoleate has increased threefold in the American diet over the past few decades due to increased soybean oil and increased corn oil in our diet. And linoleate is a major variable. So we could, if we can identify it, then we can, in principle, we can control that. Another example is with air pollution. one of the very common metabolic characteristics we see with air pollution, and I think Carmen may talk about some of Donghai Liang's work on this, but arginine is depleted, it's decreased in association with air pollution. And so that means we could, in principle, therapeutically intervene with precursors to enrich those pathways. Of course, low arginine enhances inflammation, all inflammatory processes if you have low arginine. So it's a big deal. And so I think it's really a good example.
I think it's interesting too. David Wishart always likes to point out that most of our clinical chemistry is essentially harnessing the metabolome, right? It's looking at small metabolites and much more actionable than many of the genetic sort of testing that we have. So and I think it's just kind of bringing into the, not just thinking of the endogenous molecules, but how the exogenous molecules can fit in there too. There are also questions just about the cost of some of these analyses. Like, how do you see, obviously the genome analysis has gone down tremendously in price. What point is this sort of analysis on par with what we do in genetics from a cost structure?
Okay, that's really a critical issue. That was one of the original principles, is it has to be affordable. So Doug was on the panel. Doug and I have worked on this quite a bit, and we've talked about, you and I have talked about this. The cost structure is really critical. Right now, we're at a real cost of about $600 to $700 per integrative high resolution metabolomics exposomics analysis. That would be a single gas chromatography analysis, a multiplex liquid chromatography analysis, and then something I did not really allude to, but we absolutely must include it, is ICP-MS to measure, get a broad spectrum of our metal exposures.
So another question, and kind of related to that, and thinking about what you put together, is that when you do this sort of multi-omic level analysis, and if we have proteomic data and genomic data, do we treat all of them the same? Or should there be more weight given to certain layers of that omic structure?
Well, that's a great question too. The beauty of these integrative omics methods is that you can set your filtration, you can set your thresholds wherever you want. And so that's the beauty of that hierarchical approach that Shazal Li used, is you can set your thresholds. And so if you only want to look at the top 50, the top 100, or the top 500 proteins, you can do that. Similarly on the transcripts, similarly on any of the mass spectral features. So you have that ability to shrink and expand the universe of what you're looking at. And that gives you the ability to let the data tell you what the structures are. And a good example on this is when we looked at choline and phosphatidylcholine, the lipidome. What you see with the lipidome is you will have 500 very, very highly correlated lipids. The question is, do you need to look at 500 of those? Absolute lipids is different. And the answer is no. You can cluster those. You can reduce those and use basically a surrogate for or one selected metabolite or some clustering method to get a parameter.
So, and kind of follow up to that, and thinking about the population heterogeneity that we see in this sort of data, what are the sort of approaches we can use to reduce that? And how important is that sort of inter-individual variability when you're looking at a cancer type study of a large population?
So I think from the standpoint of exposome and metabolome, I think what we have to do is to get into some of the large data sets, or large sample archives. The U.S. The Armed Services has a I guess it's Department of Defense serum repository, has 300 million samples. And those are collected from individuals across the country, collected over the past 40 years. And resources like that could really be tapped to answer the question. I wouldn't want to venture guesses as to how to do it any way other than just.
All right, well, I think that's all the time we have to answer these questions. Dean might be able to go into the Q&A box and answer some of these that we didn't get to, but Dean, thank you very much for your presentation and a lot to think about. And we will now move on to our next talk. Thank you, Dean.
Okay, thank you.
So next up, we have Carmen Marcet, who is just 100 yards or so away from Dean Jones at Emory University. And I think what we're going to hear now is something I've always been very intrigued and impressed by Carmen's work, is that when we think about multi-omic integration, we always start with that genome part. And when you go to the parts that are closest to that, like the methylation of the genome and things, I think there's more concordance and better chances of success and that link there. And so Carmen's done a lot of work in this area that's, to me, like nearer to the genome and how you use the genetic techniques, which I think have really helped advance because of this sort of connection to it. And he's also worked in some very different kind of sampling selections with placental work, which is very nice. So with that, I will turn it over to Carmen.
Great. Thank you very much, Gary. I'll share my screen here. Okay. So thanks a lot for the introduction, Gary, and for allowing me to be part of this workshop. What I'm going to do today is provide a couple of examples from the work that's been going on here at Emory in the Hercules Exposome Research Center and in my own lab, where we're bringing together environmental data and omics data. I just want to give a disclaimer up front that these are not specifically about cancer, but I think the same principles that I'll talk about for this integration will apply. And I think also including some of the caveats that were talked about during a lot of the talks yesterday. So when we think about integrating environment and omics, we can think of various layers of biological features that can be looked at. So it could be genomics or epigenomics, transcriptomics, proteomics, or metabolomics, and other features now that as we think more and more about the different layers of biology that are involved in the disease process. When we're doing this, we can think of a few approaches for integrating the environment into these biological signals. Some of these approaches might be common across these different biological features. We'll talk a little bit about meet in the middle type approaches. We can also think about causal frameworks with formal mediation tests. We can think about risk scores, and I think this came up a little bit yesterday in some discussion of genetics and Mendelian randomization. But I also think that this could be a possibility with epigenetic features. And that also came up yesterday in the discussion to think about what might the future be with using epigenetic risk scores. We could also think through how to integrate across these omic layers. And this can kind of be done in a quantitative trait analysis. So that's much more of the central dogma type approach, where a genetic feature is now leading to the change in a quantitative trait like a transcript. But we could also think about some new approaches using AI or machine learning technologies to really just bring all of this data together and kind of see how it falls out in a much more data-driven approach. And so today we'll talk about a couple of examples from these different types of integration opportunities. So I'll start with the meet in the middle approaches, which I think are becoming increasingly common in studies of the environment and biological features. So here, what we're looking for are omic features that really overlap between the exposure and the outcome after demonstrating that there is some relationship between an exposure and an outcome. So what you'll do is you'll run an XWAS, where that could be really the omic of your choice with an exposure, and then separately run that same XWAS, so looking at the outcome with all of those biologic features, and then identify which of those features sit at that middle point. So which ones overlap between these two larger groups of associations. And to give you a sort of a concrete example, I'm going to walk through the work of a talented graduate of our Emory Environmental Health doctoral program, Ron Chang, who's now at NIEHS, and led by our really innovative early career faculty member, Donghai Liang. And they were looking at metabolomic features that link PFAS exposures to birth outcomes, and particularly infant growth, in a study of African American and Black women here in Atlanta. And so for this study, sort of the first step is to identify, can you see an association between exposure and that outcome? So here they demonstrated that for specific PFAS chemicals, PFOA and PFNA specifically, were associated with both reduced birth weight on the left and with higher risk for gestational age birth on the right. So now we've completed that bottom part of the triangle where we see the relationship between the exposure and outcome. They then went on to do the two sides of the triangle. So now performing the in this case, metabolome-wide association studies, or MWAS, for the association between the exposure and all of the metabolites in serum, and then for the outcome and all of the metabolites. And they identified a number of specific metabolites that were associated with these exposures and then with the outcome of interest. So in this case, I'm highlighting one of these findings for uric acid, where they identified that uric acid was increasing with exposures to PFOA or PFNA, but that as that uric acid increased, there was reduced birth weight and an increased risk for gestational age birth. So this kind of starts linking these pieces together, so that as these PFAS chemicals are increasing, uric acid is increasing and fetal growth is decreasing. So really they found a number of these different metabolites that were associated with PFAS and growth, including amino acids, lipid and fatty acids, androgenic hormones, and uric acid, as we've seen. And they can go on to use these features to think about some of the pathways that might be involved. And there they were able to identify that pathways like oxidative stress, inflammation, and placental transport seem to be important mediators of these environmental stressors. And I think what's important there is that's the kind of things that could become actionable, is trying to identify where some of these pathways are. And that might be the way we could think about where interventions could be really helpful. Now, to give you another example of this type of approach with a little bit of a different flavor, I'm going to highlight work from our group that was led by Maya Dyson-Roth, who's at Columbia University. And this is where we were integrating data from multiple metals exposures with transcriptomic features in a placenta and looking at birth weight. So if we look across a panel of 16 trace metals, we do see that there are some, like arsenic, cadmium, and tin, that individually, just in basic models, are associated with a risk for gestational age birth. We want to ask, since in reality, no one's exposed to any of these in isolation. It's really, we need to take a much more holistic approach. What about the mixture of these metals? So using a weighted quantile sums approach, we were able to identify a mixture of metals, which as it increases is associated with SGA risk. And that arsenic and cadmium were really the predominant metals that were driving that association. And so we've established now that bottom part of the arrow, that exposure to outcome relationship with this mixture of metals in this case. Then to examine the transcriptome, we performed a dimensionality reduction using weighted gene correlation network analysis. The idea of this is to identify groups of co-regulated genes, and then using a pathway analysis, we can understand what kind of functions do those co-regulated genes operate in. And you can see on the left, some of the functions that we identified with these co-regulated modules, like cellular respiration or vascular development, real functions that we would expect the placenta to be playing as part of fetal development. So we have these modules now of expression. We have the weighted quantile sum, so we can now start bringing those pieces together. So by integrating the data, we can relate the SGA associated WQS index, so that mixture of metals, to the eigengene values that come from the weighted gene correlation network. And we identify two modules, one related to gonadotropin and glucagon synthesis and one to gene expression, that are related to both exposures and the SGA risk. So this is now looking at those two sides of the triangle and putting those pieces together. How does the environment relate to these signatures, and how do these signatures relate to the outcomes? And so taking it together, we identify that as we see increases in this index of a mixture of metals predominated by arsenic and cadmium, we see decreases in pathways representing gene expression and increases in pathways representing gonadotropin and glucagon, and those are related to then decreases in birth weight. And then for one last example of what I think could be really promising approaches to integrating the environment with some of this omic data, I want to touch on the potential for using these types of risk scores. And I think this could be really important even for studies of cancer, as it may be a way to start thinking through the limitations of having to use retrospective study designs, because this may open up opportunities for being able to assess exposures in the past, even if we're looking at it in a retrospective format. So in these types of studies, a risk score is developed either using a large data set that's already been published and that the results are available, or taking a data set and splitting it into a training and a testing set, and then performing your analysis. So you're looking at a phenotype or an exposure, along with then all of the SNPs if you're doing a genetic study, or DNA methylation if you're looking at epigenetics, to identify all the SNPs in some cases, or sites of DNA methylation that are associated with either that phenotype or exposure. Then you use that data to estimate weights from the summary statistics from all of those results, to build a risk score that then can be applied to new studies. And so you're basically making use of existing data to build a risk score to apply to a new set of data. And I think this holds opportunity both to provide us some information about historical exposures, also potentially for predictive, prognostic, or diagnostic approaches, I think, as well. And I think that's where people have been thinking about some of the genetic risk scores, but I think epigenetic risk scores can hold some of those same features. And just as an example of the potential for this to work, I want to show some work from a very talented early career faculty here at Emory, Anke Hoels, who has been looking at developing these types of epigenetic risk scores. And in this case, she published this study where she was looking at prenatal tobacco and alcohol exposures. And can she identify risk scores to underlie what those might look like? And I thought this was a great example of how we can make use of a lot of the existing data that's out there. She used results from the PACE Consortium meta-analysis. So this is a large meta-analysis of studies of prenatal smoke exposure and cord blood DNA methylation. In this case, it was across 9 cohorts and almost 6,000 newborns, with nearly 900 exposed to prenatal tobacco use. So this was published data, published summary statistics from that data. Anke was then able to derive a risk score based on those summary statistics, using those to estimate weights for her risk score, and then to apply that risk score to a new set of DNA methylation data in the South African Drakenstein Health Study cohort, which has 262 children with cord blood DNA methylation. And then she has the maternal biomarker data on cotinine. So then she can relate this risk score to a more objective measure of prenatal tobacco exposure. And just to show you that that works, right? This whole concept is reasonable. This is relating those smoking-related risk scores and looking at it within non or passive smokers versus active smokers, and that was determined empirically from that cotinine data. And so you can see that these risk scores are increased within the smoke-exposed children. So I do think that there is some real promise here. Now, she went on in this manuscript to examine if this could be used then as a mediator to relate prenatal tobacco or alcohol exposures to some neurodevelopmental outcomes, and it wasn't as successful in that part of the question. So we have to think about how we actually make use of some of these, and if they're really appropriate as a mediator, or if really they're just appropriate as a risk marker or an exposure marker, and the best way to apply them to studies. But I do think that there is some real opportunity to be able to make use of this type of data and these types of score approaches. And I can imagine these being really useful when we're thinking now about some of these large cohort studies that we have where maybe this data wasn't collected, right? And so how can we go back and try to assess some of these different exposures when the data wasn't collected, in the past when it really needed to be? So now there are some limitations to a lot of these integrative measures. We have to think about the temporality, and that tends to be a real challenge in sort of all studies. We tend to be measuring exposures and these biological features at the same time as maybe disease onset or same time as outcome onset. And so really being able to get past some of those temporality challenges to really get at causal features is going to be a challenge for these approaches. As Dean just mentioned, thinking about, are we able to assess these things in target tissues? And often we can't, right? And I think we're thinking more often about accessible tissues versus target tissues. But I think there can be reasonable biology understood by examining even accessible tissues. If we're looking at blood, we're looking at the immune system. And so we're now asking, how are exposures affecting the immune system to now affect a disease process? And so as long as we understand what we're looking at and really interpret it appropriately, I think that there's real possibility. At the same time, by being able to make use of target tissues, we could really understand direct effects of exposures on these disease process. And in cancer, that might be there may be more opportunity to be able to do that than we might see in some other disease. Again, we have to think about how do we start incorporating all these different layers together using these dogma-driven or agnostic approaches. And I think the question is still out there, is if AI approaches can be used, how do we really apply those? Do we take into account some features as more important than others, and how do we weight that? So I think all those questions are going to have to be brought up to really understand how to make use of this. But I do think that there could be possibility there, and really to demonstrate some of the utility of this. At the same time, I do think that there are a lot of promises of omic integration. I think it really has an opportunity to inform mechanisms that underlie the link between the environment and health, and really identify places for targeted intervention. So we're thinking about precision environmental health and precision approaches to try to address environmental exposures. These may be able to help us to identify some of those. I also think that they're useful for the more holistic exposome examinations. We may be able to start thinking about how to integrate across what Dean talked about using the metabolomic studies and exposomics, then with some of these other features like epigenetics, to really be able to bring together some of that data. And that may also be a way that we can overcome some of the costs. If we can utilize existing data to identify some of these risk markers that then are at a lower cost able to be applied to large populations, we might be able to still move the field forward and really inform on different exposure histories. So I will stop there. I'm happy to take any questions.
Okay. Thank you, Carmen. A couple comments I have in kind of thinking about what you were saying is this idea of the accessible tissues. And so I think a couple studies that are very interesting is the, if I get the name right, the TAC trial, the one that they're looking at peripheral tissues, central sort of markers in animal models to see kind of what you're seeing with that. But another, really exciting area is the use of extracellular vesicles, where you have organ-derived, liver-specific, brain-specific extracellular vesicles, which is providing you this information of like that source of where it's coming from. And I think that the integration of some of those tools, I think, could be very helpful to get to that sort of organ-specific aspect of it, which I think is very exciting. Also on this, the point of this more holistic approach, I'm starting to see data come out of some of the larger biobank cohort studies where some surrogate of a exposome, exposure risk score sort of thing, can be as powerful as a polygenic risk score, for many diseases. And I think that that's something where, the work that you've done, the work that Dean's done, looking very much at these sort of biological markers, we don't typically work with the people that are doing satellite imaging or these other sort of, very external parts of it. And I think it's one of the things that we can address too in our panel discussion is how to bridge those. How do we bring those very different communities together to get all of that sort of external information? And I know that you've done some of this work at Emory, but kind of what are your thoughts on kind of bringing some of these geospatial approaches closer into the sort of biochemical and genetic sort of approaches?
Yeah, I mean, I feel like if we think about air pollution as an example, I mean, there it's being done, right? And I think that's being done a lot. We've done a lot of studies on air pollution, metabolomics, epigenetics, and some of the extracellular vesicles, where that's derived from these approaches, these remote sensing or these sort of model-based approaches. And I think there is a real interest in bringing in a lot of the other data that can be obtained from those kind of sort of place-based assessments. And so I think people are moving in that direction. I think what it is you have, I think, as you said, there's just different researchers who are working on these questions. And so it's really being able to bring people together and have that communication, develop some kind of shared language around it so that people understand what they're talking about, and the other challenge with it is getting a better understanding of how do we incorporate the error that's involved with some of those place-based exposures that we might see less of when we're doing, direct assessments of an exposure, right? So if you're measuring burden of, some chemical in the body, well, we understand kind of what the error is on that, and then how that might relate to some of these biological features. Estimating it based on place, there's a lot more error around that. And so we're going to have a fuzzier association that we have to think about. And so how do we incorporate that, and then how do we propagate that so that we can really make some good inference, downstream, I think is going to be really important.
Thinking more about some of the epigenetic sort of marks and some of the data that you showed from looking at past smoking history and things is, this is like kind of like in comparing the sort of biological data to the epidemiological data is with smoking is that people's recall of their history is pretty accurate, right? They know how many packs they smoke, whatever it would be. And then for other aspects, it's less accurate.
Right.
And like the idea is like, how can we use, again, it's a fusion of these data sets, right? The idea is that how much of the sort of classical epidemiological survey instrumentation needs sort of biological validation, because the idea is that if it works, then you could say, well, we can totally trust the survey data, or the epigenetic sort of mark might give you a better reflection of it. Like, how do you, again, because it's something where I think both sides have something to lose. And so they may not want to learn the limitations of it, but it seems like from a field standpoint, like for the cancer community, if they knew that this sort of blood test could tell them all these key things in populations they don't have, or that they should totally rely on this sort of survey instrument. So what are your thoughts on that?
Yeah, I would say smoking has worked really well, and I think you're completely right, because people are good at assessing it, and it's kind of like a hammer, right? I mean, people use smoking always as a model exposure, right? Model exposure for air pollution, because it's a big exposure. It's a complicated exposure too, but we tend to see biological response to it. I think that is a big challenge, because I think when we're thinking about some of these other types of exposures, one, I think you're right, the assessment is going to be challenging. It's hard to ask a person, how much PFAS do you think you've been exposed to? That's not something that people can get their head around, because we don't even know where it's coming from all the time. So I feel like for some of those types of exposures, then there's real opportunity in identifying some of the biological markers that are going to be able to tell us, do a better job about assessing that kind of exposure. And I think that might be true even for even social exposures, right? When we're thinking about psychosocial stressors and things like that, there might be a real place. Yes, you can use questionnaires to ask people about sort of their experiences, but every person experiences them differently and for a lot of different reasons. Getting to a biological marker of sort of that downstream effect might be more useful, because it's going to integrate the exposure and the way that a person experiences that exposure. And that could be true for chemical exposures and other types of exposures as well. But it's much more of an integrated measure, instead of assuming that the way I say that I'm under a certain level of stress, and that's equal to the same level of stress that you would say, but maybe it's not, and maybe we perceive it differently because of other things. And so it's being able to take a lot of those different pieces into account.
Yeah, it's interesting thinking about the sort of social aspects, and just a tension I've observed in the field is the people who study things from a more sociological construct have an aversion to over molecularizing everything, right? The idea is that these are complicated social constructs that may not be able to get down to a molecular level. But if these social constructs are having an impact on health, there does need to be a biochemical signal. Like you said, this is where that integration can come in. And so it's another place where I see these sort of the partnership being very critical, is establishing the relationships with the social scientists who have collected that qualitative data, this very rich sort of data, that can have the conversation with someone who can measure more of the molecular pathways, but also respecting that it's, that it's something where it's not, you just can't bring everything down to this molecular level and just say that we can have a biological map of structural racism. But the idea that if these sort of structures are influencing health, there does need to be this biological pathway that's disrupted in order to do that. And so to me, it gets back to the sort of really not just thinking like in a multi-omic standpoint, like we tend to think on this omic layer, but how you really bring in more of those social constructs into it, which I think the exposome sort of model has wanted to do, but it's been one of the harder areas to get through. So I don't know how much experience you've had working with people that work on more of the sort of sociological aspects, but any thoughts or comments you have on that?
I mean, I actually think that there's real opportunity by being able to demonstrate some of those biological downstream effects to prove that these are truly, causal, agents in disease process, right? If we really want to demonstrate that these have an effect, we can find the biological, underpinnings of them. And that to me seems like that could even help the argument more so. And so, I feel like it could be win-win, for doing that. And I do actually think that people are starting to embrace it. I mean, it's something that we see here. The whole concept of the exposome, seems like this very, academic discussion. How do we get this holistic measure? Well, when we meet with our community boards here, people love the concept of the exposome because they actually finally feel like it's listening to them and not just focusing on, some chemicals that scientists want to focus on, but also is thinking about the rest of their life, right? And how these other pieces fit into that. So we found that people really embrace the idea of it and really think about that holistic piece of it. So I do think that there are ways to use some of that, together and really kind of build better cases for things.
So in thinking about this idea of like developing an exposure risk score, and we could take, mental stress and psychiatric disorders as an example, is what sort of study or studies do you think would be necessary to develop the reference panel for that? Like to say, if you had to pick a few of those, what sort of scale do you think we have to be at for that to work?
I mean, the challenge is it has to be big. I mean, I really think we need sort of large studies that have enough variability that we can really pin down some of these relationships. I mean, the reason the smoking stuff has worked so well is because it's also been done in thousands of people. And so we can feel really confident about what we're identifying. I think the GWAS are similar, right? So you can very confidently identify all the variation associated with some outcome and then use that in another way. So I do think that there has to be sort of these reference data sets out there. I'm not sure they have to be in the context of a specific study. I mean, they may be something where we could be using, like large hospital data and samples that are available and integrating those pieces together. The all of us kind of cohort piece where instead of trying to identify everything related to some outcome, just kind of broadly characterize all of this and allow then for that, those kind of relationships to be identified, more specifically as they're needed. That might be the way to go. So you sort of need really a broad population type assessment.
I think this is like where, for example, in something like All of Us and UK Biobank is, I think this gets to the idea is that, we can explain how we can do a very good genomic analysis in those studies, and so they tend to get funded. But when we start talking about complex environmental exposures and sensors and all this complexity, I think we often scare them off because they say, how can you do that? Because you don't have standard technology and things. And I think this is something that we need to address because we're competing for resources against people that can just sequence sort of thing. And so, is it something where we need to be thinking more about a focused exposome panel that could be standardized versus our discovery sort of phase where we're looking at all these different things? I noticed Dean also put in the chat, which is interesting about, we talk about how primary smoking tends to be a pretty accurate indicator of like the dose of smoking. But if you look in children and look at secondhand sort of exposures, the self-reporting isn't as effective, so it is something when the dose is lower, you get more variability in there. And that's something that we've seen like in looking at other studies where things like coffee consumption are shown to influence a disease. And we can go in and measure dozens of metabolites of the coffee things that it's a much richer data set when you can bring that biochemistry into it as well. So So I think we have a lot of work to do. Keep doing your work because it's going to be very critical in this. But it is now time for us to move to our panel. So thank you, Carmen, and we will move on to the panel discussion now. I believe what we have here is each of the panelists, which hopefully they'll be pinned here pretty soon, and we can get them lined up. But we will be hearing a few-minute intro from each of our panelists. We have Scarlett Gomez from UCSF, Tom Metz from PNNL, Doug Walker also from Emory University, Ivana Young from University of Colorado, and Nat Rothman from NCI. So I am going to go in that order and start with Scarlett.
Great. Thank you so much. And thank you to the organizers for giving me this opportunity to participate in this, what's really shaping up to be a super interesting workshop. So in my 5 minutes, I really wanted to focus on this topic that we've started to touch upon, which are the structural and social drivers of health. As we know, upstream social inequities create the isms, that is racism, sexism, classism, etc., by which policies and institutions can be biased against certain population groups in this country. These structural isms are considered the fundamental drivers of health inequities. They shape and produce the social determinants or drivers of health. And so just as we've heard, we're abound in a sea of riches where it comes to wealth of omics data. There's also an increasing wealth of geospatially referenced data for characterizing these structural and social determinants or drivers of health. There are, of course, data that, as I'm sure we all know about, from census, American Community Survey, that can be used to characterize a broad array of social and demographic neighborhood factors. Street reference maps and business and licensing data can be used to characterize walkability, built environmental factors. And more recently, researchers have used data from Home Mortgage Disclosure Act, or HMDA, to derive measures of contemporary redlining and racial bias in mortgage lending. And these have been integrated into several recent studies, such as our response study of prostate cancer in African-American men. Even more recently, researchers have begun to use the 1930s Homeowners Loan Corporation, or the HOLC maps, to associate patterns of historical redlining to current day cancer disparities. At the same time, we have emerging approaches like machine learning and image processing, advances that have enabled us to characterize environmental features like green space, blue space, and light at night. These are features that our group has been using, for example, integrating into the pathways study of the breast cancer survivors. We and others have innovated an approach to conduct a virtual street audits using Google Street View, and several colleagues have used georeferenced Twitter data to characterize temporal and geographic trends and racial animus, another potential source of geographically referenced social environmental data. We've also begun to develop better data and approaches to measure residential history and changes in or cumulative exposures to environmental stressors over time, another important topic that we've been discussing. In addition to recognizing that neighborhoods are dynamic and phenomena like gentrification, displacement, and persistent poverty and disinvestment are important to capture and assess for their impacts on cancer. There was a recent geospatial workshop last month that was co-hosted by USC and NCI, and it was really, truly amazing to me to see how far we have come in just the past 5 years in our ability to broadly capture such a wealth of environmental data. However, indeed, we have an increasing problem of riches, a good problem, but one that requires better tools in our toolbox. And so to get us started, I'd like to propose 4 ideas to help us think about how we can use this growing wealth of environmental data appropriately, responsibly, and also for the greatest impact. First, with so much data, and some of it is fairly easily and freely obtainable, it's really easy to get carried away. I can personally attest to this. So I would propose that we should always aim to use conceptual frameworks and models to guide which domains and dimensions of structural, social, and built environmental data is of the greatest relevance and interest for the outcomes and omics that we're studying. What makes sense as upstream drivers of the omics that we're focusing on. Conceptual and causal frameworks can help us to determine which environmental factors precede and cause another environmental or individual level factor. Second, we need to carefully consider which environmental factors interact with one another and produce synergistic effects, or perhaps act in concert to create a broad profile of effects. Make sure modeling approaches such as latent class analyses and modern Bayesian and spatial adaptations are emerging approaches that may help us to sort through some of these interactions among multiple environmental components. I think this is definitely a ripe area for further developments, but we also need the tools to be broadly accessible. Third, and we touched upon this yesterday, but it's so critical for multiple disciplines to come together to the table, as we've been talking about also in this session. Of course, in this workshop, we're talking about integrating Big E with omics. And so this requires those who work with the Big E to collaborate with those who research the omics, but it really takes a lot more than that. Of course, geographers are essential, but I've also learned that we also need demographers, sociologists, economists, geostatisticians, bioinformaticians, and the list goes on to work with us all to develop the tools for these toolboxes. I'd also like to note that we, or at least I, have not been properly trained on how to do multidisciplinary team science well, and we really need to focus on training our trainees and how to do this in a much better way. And finally, and perhaps the most important consideration, and we also touched upon this earlier as well, and I really appreciate Carmen's comments about this, is that we need to engage with and listen to the communities that we're studying and listen to their life experiences. Let them guide us to help us to interpret our results and make sense of our data. Thank you.
Thank you, Charlotte. Yeah, I think it is something that we've seen through the community engagement cores that many of our centers have. This has been a key part of that, and it's something that it's great to see that these institutes value this. So now let's go to Tom for comments on your side.
Yeah, thanks, Gary. And would like to thank the organizers for the workshop and the great lineup of speakers. To put my comments in a bit of context, I'm having connection issues here.
We can hear you fine, but
Okay, let me turn off my camera and see if that helps. Hopefully I'm still with you.
yes, I can hear you.
Yeah. So just to put my comments in context, our laboratory focuses on the development of mass spectrometry based methods development, technology development, methods development, and their applications in systems biology studies of disease, not one disease in particular. And my particular background in that regard is applying these methods in the study of infectious disease and in type 1 diabetes. I really want to riff off of some of the data and comments that Dean made in his presentation regarding the technologies and the knowledge gained from metabolomic studies, and particularly using the analytical techniques that we have available to us these days. There's really a diversity in analytical techniques for metabolomics, NMR, gas chromatography, mass spectrometry, liquid chromatography, mass spectrometry. And there's really a diversity of methods employed here. And it speaks to the fact, I believe, that the community has not yet developed the leading technology that would allow for sequencing metabolome in an analogous manner as what we do for, say, gene sequencing. It really has resulted in this lack of harmonization of methods across the board. And I feel that it's really an opportunity for the community, all of us, researchers and funding agencies as well, to have somewhat of a moonshot project. Dean mentioned the Human Exposome Atlas. Another way to think of that would be the Human Metabolome Project that might require a similar investment as what was put forth into the Human Genome Project, which was $3 billion in today's money, and about 15 years of work that really revolutionized the field in that regard, leading to the sequencing technologies that we have, the informatics technologies that we have now, that really make sequencing more or less a commodity relative to the effort that goes forth into metabolomics type measurements. Also, these instruments are so exquisitely sensitive. Dean mentioned often you can detect up to 1 million signals. After dereplication, that will reduce down to some number of actual molecules. And then the percentage of those molecules that we can actually ascribe chemical structures to in order to understand what they are and from where they're coming, it's really a small number relative to the other quantity of data that's collected. And so a a concerted effort by the community to solve this challenge of how do we deal with all of this chemistry effectively and efficiently in a single measurement. I think it's really worthwhile and will lead us to new insights and balance out that equation of G times E when the volume of data of G is much larger usually than the volume of data on the E side. Thank you.
Okay, thank you, Tom. So following up on some of our mass spec based insight, I'll turn it over to Doug Walker, who I'm sure will mention something about that.
Yeah, so again, I would also like to thank the organizers for the opportunity to participate in the workshop. This has been a fantastic past day and a half, and we've been discussing some very exciting topics. Similar to Tom, my background is in the actual lab side of developing these approaches that allow us to characterize the human metabolome. One of the differences in the approaches we've taken is we really have tried to systematically optimize and emphasize the sensitivity of our methods for specifically detecting these environmental biomarkers or exogenous compounds that are originating outside of the body. And we've approached that through a number of different strategies. The first being, really trying to diversify our analytical platforms, as Tom mentioned, so we're not necessarily biasing ourselves towards one class of chemicals, but really trying to detect as wide of a range of chemical space as possible, so we have the ability to screen for many of those environmental biomarkers. The other challenge we face in these exposome analytical methods is that there is a very wide range of diversity in exposures over a lifetime. So it can become very challenging to think of a reference exposome panel when we realize that the diversity of exposures that occur across different geographical regions can vary significantly, both spatially and temporally. And so the way we've approached that challenge is through really trying to develop methods that allow us to maximize our throughput and provide analytical strategies that actually allow us to perform these exposome measurements in large populations. A lot of that has been through incorporating the principles that Dean mentioned in his presentation. So moving from manual to fully automated sample preparation methods, establishing robust, reproducible, and high throughput analytical methods, and also developing the informatics that allow us to take full advantage of that data that we're generating. So this has allowed us to, basically establish a strategy that provides, high throughput analysis of biological samples to characterize the exposome, which is moving us onto or into a scale that's more consistent with what's being applied for GWAS and other larger omics studies. And so that ability to really maximize our N gives us an improved power for detecting, both interactions and relatively low effect size associations with outcomes, as well as capturing that diversity of exposures across different populations. I completely agree with Tom's comment in that one of the ways that we've essentially failed as a research community is to really try and identify what our standards are and have a cohesive definition of the exposome that we're trying to incorporate into our studies. If you look across the literature where the exposome is being defined, it can include anything from measuring 100 chemicals to thousands of chemicals to considering external exposures. And so I think we need some consensus on what it means to operationalize the exposome within a research study. And there's a number of initiatives underway at NIEHS and some of the other institutes that are really trying to emphasize that and improve that understanding. I think another area, and this follows naturally from this concept of standardization, is how can we start creating cumulative resources for exposome measurements? So we have talked about this idea of a cancer exposome atlas. So how can we start building the framework where we stop looking at studies as these isolated entities where we run one study, perform our analysis, and that's the outcome, and rather look at individual studies as one piece of a whole, where we're combining across different cancer studies, across different diseases, and looking at, essentially exposome-defined differences among these diseases. And a lot of this will come from standardization. There's also a lot of efforts to improve our consistency in ontology and identify what is a acceptable study to be incorporated into these cumulative resources. But I think that's also an area where we really need to be thinking and trying to leverage our resources so we can build these cumulative databases that allow us to look at exposures across populations and across diseases.
Thank you, Doug. Okay, so comments now from Ivana.
Thank you so much for the opportunity to briefly share some ideas that we've had on integrating exposures with omics in interstitial lung disease. So I'm going to spend the first couple of minutes just introducing the diseases we studied to give context, and then tell you about a couple of ideas we've had. So ILDs are a group of disorders that cause progressive scarring of lung tissue. And once this scarring occurs, it's generally irreversible. Treatment options are limited. There are ILDs that have strong genetic causes, but many others are caused by long-term exposures to occupational hazards such as asbestos, coal dust, beryllium, etc. And relevant to this workshop, it's worth noting that ILD is associated with an increased risk of lung cancer, even after accounting for confounders, and to have a poorer prognosis than lung cancer without ILD. So in collaboration with Lisa Meyer, who is an occupational lung disease specialist at National Jewish Health, down the street from us in Denver, we have ongoing studies of two related ILDs that are characterized by granulomatous inflammation in the lung. One is chronic beryllium disease, or CBD, and another one is sarcoidosis. Both of these diseases start when antigen is taken up by antigen-presenting cells, which then leads to binding of self-peptide. And then these antigen-presenting cells present the antigenic compound to CD4 positive T cells, which leads to release of inflammatory cytokines and development of granulomatous inflammation. Some individuals progress to lung fibrosis while others don't. And what we study is epigenetics as a mechanism that may account for some of these differences in response to the same level of occupational exposure in genetically susceptible individuals. So in chronic beryllium disease, it's simpler. We know the antigen, it's beryllium, which is also a known carcinogen, and we know the key genetic risk factor. Sarcoidosis and some other ILDs are much more complex in terms of multiple antigens, so multiple exposures, and multiple genetic risk factors. And these antigens and genetic risk factors vary geographically and across different ancestries. So as part of our work, what we've tried to do is use cohorts that Lisa Meyer and her colleagues have collected and integrate occupational beryllium exposure data with epigenomic profiles, mainly DNA methylation, but we've done some attack sequencing as well, and transcriptomic profiles of lung inflammatory cells. And we get these lung inflammatory cells by bronchoalveolar lavage from chronic beryllium disease patients, as well as precursor, which is beryllium sensitization, and then we can compare them. One major limitation that we've noted in this work over the past few years is, these are relatively rare diseases, and so we have small cohort sizes. So maybe this is similar to some studies of rare cancers or maybe younger individuals that we discussed yesterday. Another limitation has been confounding by differences in proportions of different cell types. So for example, usually CBD individuals have more lymphocytes than sensitized individuals, or we have noted differences in macrophage subpopulations. So because of these limitations, we have turned to ex vivo models of exposure of these lung inflammatory cells to beryllium. And so in one of our currently funded studies through NIHS, what we're doing is we're characterizing time course of lung immune cell response to beryllium exposure in these ex vivo models. So we're using single cell attack sequencing to look at chromatin accessibility and site sequencing to measure gene expression as well as protein epitopes. We're doing this in cell culture ex vivo at 3 to 4 different time points following treatment with beryllium. And then what we're doing analytically is performing integrative trajectory analysis of these single cell omic data sets to identify key networks of genes and drivers of cellular response to beryllium. And again, this is all now cell-specific manner. So our goal is that once we identify these key networks and drivers, that we can then go back to our existing cohorts and perform more targeted analysis. So our hope is that this approach will overcome both limitations of small cohorts and confounding by differences in cell proportions. And then more importantly, we hope that what we learn in chronic beryllium disease, which is relatively simple, can be applied to sarcoidosis and other more complex diseases that have multiple antigen and genetic risk factors, and we think fit this paradigm of multiple exposures or exposome, as we've been discussing in this workshop. Thank you.
Thank you, Ilana. Matt Rothman, you're up next.
Hi there. I'd like to echo what my colleagues have said about being very grateful to the meeting organizers. It's been a great workshop, and I'm very pleased to be able to speak for a few minutes about some work we're doing and tie into a few of the themes that have been discussed. I want to speak about at least one approach to collecting information that could be used for a reference panel or environmental atlas ideas that have come up over the last 2 days and that Gary also spoke about. And I want to talk a little bit about work that Dr. Chinglan and I have been doing over the last 20 years. We are occupational environmental epidemiologists who do a lot of work in molecular epi, a term that predates some exposomics, but conceptually is sort of more or less the same idea. We began conducting studies of populations with very well-defined exposures to unknown or likely carcinogens. And we studied these exposures in multiple countries. We studied populations exposed to a wide range, some at relatively high levels. We spent years identifying the populations so that the exposures were relatively homogeneous, or if mixtures, at least they weren't exposed to multiple different classes of mixtures. We often spent several years characterizing the exposures and then collected biological samples and processed them in many different ways, assuming, hoping, expecting, as was the case, that technologies would evolve over the subsequent 2 decades that could be applied. And what we've managed to do is to assemble a series of studies now, about 24 or so different kinds of chemicals and particulates, compounds like benzene, trichloroethylene, formaldehyde, diesel engine exhaust, different types of organochlorines, coal combustion, biomass, etc. Different types of indoor pollution, air pollution and outdoor air pollution, and used relatively standardized protocols to collect samples and process them in many different ways and store them as best as we could, imagining what would be the requirements of future technologies. And we were lucky in that the basic approaches to storing samples really haven't changed that much, and these samples seem to be amenable to analysis through multiple types of platforms. And what we are now doing is going through fairly systematically about a half and different platforms on these perturbations that we see with different types of omics platforms within a chemical, to compare across different types of omic platforms and to compare across chemicals and then across classes of chemicals. And we hope that by focusing on very well-defined populations based upon exposure, and unequivocally understanding their exposure, that at least we can answer one part of this complex question, which is, if you are exposed on this day to this amount of compound and had this type of cumulative exposure and this type of pattern over the years, what do you see? And what do you see on platform A, B, C, D? How do they relate to each other? How do they relate to other kinds of chemicals within class and across? The second thing that we're trying, and we think that this paradigm, while it's very important to sample from the general population, that this paradigm can also be used to identify and study populations exposed to other kinds of, whether it's chemicals or particulates or whether it's social stressors, but basically identifying populations a priori that have a wide range of exposures. And you don't need to study many people. What you do need to do is you need to study people who are selected and very well characterized based upon the exposure that you're interested in studying. And I'm sure there are, as far as social, psychosocial stresses, and as far as people who are the source of those stressors, as well as people who receive those stressors, various populations in the US that one, for example, that one could study to try to begin trying to understand the biological impact of psychosocial stressors. So this is one approach. How do we build up, whether we call it an environmental atlas? Ching and I described this as a library years ago, sort of using a molecular paradigm. How do we build these up so that we can interpret the second part of what we and many others are doing, which is, one, after looking at the biological imprints that these exposures have, which is valuable in and of itself to understand the underlying mechanism, to then ask the question, do we see similar perturbations with more or less some level of specificity for the exposure if we now begin looking at disease, cancer, let's say, for example. And we've taken a number, and we're still starting almost at the beginning of this process. We've done a good bit of work with Dean and Doug in this area, where we look at results of cross-sectional studies of populations exposed to different types of air pollutants, and then ask the question, do we see certain metabolomic patterns that are linked with air pollution? Do we see, are they associated with the risk of developing lung cancer prospectively? And we've done this with metabolomics, we've done this with methylation. A number of scientists in our group, Mohammed Remen and Tel Blechter, have some really nice work showing exposure, biomarker relationships, and then biomarker cancer relationships in prospective studies, particularly in some cohorts of non-smokers. The idea, the so-called idea of meeting in the middle. So we think that ultimately, this approach that characterizes very well-defined groups of people based upon exposure can, while not the only approach, can contribute to developing a library of exposure marker relationships that can help us in interpreting what in the future will be a plethora of prospective studies looking at these platforms and trying to link them to actual disease risk. Thank you.
So a couple of things kind of stand out from what you just said is that the importance of standard methodologies, right? You know, if these things weren't collected in such a way, you couldn't go back and interpret the data. And also the value of biobanking. You know, the idea is that when it's something we'll have a study, we'll have some money to fund it, and then we have these biobank samples, but it may be very difficult to maintain them. But if they are collected correctly under the right conditions, they are of great value to us because of that. I think those are some great examples from that. And onto that point, Carrie put into the chat the RFA around data and meta standards for environmental health sciences. And I think this is a lot of what we're talking about is, while it was a simpler system, the genomics community pretty quickly came around some methodology and terminology that helped their data to become much more interoperable from a fair standard standpoint. And I think this is something that environmental health and exposed omics need to really think about, is that if we want the data to be valuable, we have to come up with that. And so this makes me think about some of the comments on the mass spectrometry. We have these very elegant and fancy and sensitive approaches to measure all sorts of things by mass spectrometry, but will mass spectrometry ever get us to where we need to be? Like, is this something where should we invest millions and millions of dollars in doing more of that, or should we invest $1 billion in something better? And Tom, I'll let you, your hand came up quickly. I was leaning towards you. But to think about the idea is that what is it that we need? You know, what is the ultimate goal? And I think that we'll see the need for discovery in some places, but what is the deliverable that we need? And is it something that MS can get us there? So Tom, I'll let you comment on that.
Yeah, hopefully I can have both video and audio on at the same time. My connection's been a little bit unstable today. You know, I really like mass spectrometry. I grew up using mass spectrometry in my academic and research career, but at least for small molecules, I've always felt it's a little bit of a round peg in a square hole. It's pretty close to getting us exactly what we need, but it's not perfect in the sense that we can take any sample and identify everything that's in there and have absolute quantitative values for everything that's in there. You know, there are a lot of biases with the different methodologies that are used. And so it's, you know, it's close, but I think at this point in where we are with omics research, it's worth a step back. And to your point, Gary, critically thinking about exactly what we need from a particular measurement in terms of the output, and then trying to reverse engineer the technology that's used to get us there.
Yeah, and so you're in a location, you know, being in one of the national laboratories that does have the, I guess part of the charge is to innovate, right? Like to do things that are like that. What sort of push would be needed to say there'd be a commitment to do this sort of thing? Like obviously, you know, the role of like DOE with the Human Genome Project.
Yeah.
You know, if you put all of your engineers and physicists and scientists together on it, could they come up with this?
Yeah. So you know, interestingly, I've been doing a lot of reading.
Oh, we just lost Tom. He was about to give us the solution and we lost him. So other comments from those of you that, oh, there, Tom, you're back. Okay, you said you've been doing a lot of thinking and then we lost you.
Let me try and say this quickly so I can get it out before I crash again. So, you know, I've been doing some reading on the workshop reports that came out from the 1985 and 1986 workshops that were held, that were some of the stimuli that led to the Human Genome Project. And really, it was getting the leaders in the community together to really think through critically of the challenges and the opportunities in setting up a project as big as the Human Genome Project that ultimately had the goal of sequencing the entire human genome. We haven't really done that in, say, the metabolomics or mass spectrometry community. We're kind of, you know, we come together in conferences, workshops, we, you know, have our discussions, and then we go back into our laboratory and continue to implement and improve upon our own methods in our own laboratories. And that, you know, there's so many differences between methods and laboratories. I feel like we really need to come together as a community and have one large effort, say a directed effort, to achieving exactly what was achieved with the Human Genome Project back in the 80s and 90s.
And this is something that NIEHS has been holding, you know, the Exposomics sort of workshops, and their strategic plan as an institute is underway. You know, it is something that, you know, if NIH wants to really address complex environmental exposures, that this sort of charge may be necessary. Dean, your comments.
Yeah, I just want to remind Tom that when they launched the Human Genome Project, they did not have technology to do it. So it's on your shoulders to deliver us that technology.
Yeah. Well, I would love to contribute to that effort. But the Human Genome Project was something like 20 different laboratories, multinational effort. You know, I will play the part that I can play intellectually, but it will take all of us.
Well, I agree with you. It's going to take all of us, and it is going to take some creative, some innovation, because as good as mass spectrometry as I developed I developed some slides to actually address that, and I would guess that we're approximately 2 orders of magnitude away from being able to deliver what we need to be able to deliver.
Yeah.
And we may be a little bit better than that, but not much better than that.
Mm-hmm.
So you're exactly right, Tom. We have to have some new technologies that are really going to drive it forward. And we're going to have the harmonization effort to coincide with that.
Yeah, yeah.
Nat has his hand up.
Yeah, I've always been very optimistic that if we build it, if we design it right, that the analytical technology will evolve over time. So not that I think that the analysis that you're discussing is at all trivial. I understand from having collaborated with a number of you how challenging it is. But I think it's very important that we understand the other side of this equation, which is if we don't have the right study populations, that even with the ultimate revealing technology that can be used to analyze samples, if we don't have the right samples that haven't been characterized correctly, that there's a limitation to the yield from the enormous amount of effort that will go in, that has and will go into developing these analytic methods. So I'd like to say that it seems to me that there's sort of a catch-as-catch-can silo approach that has existed in the world when one thinks about, well, what are the exposures that we need to study? What are the exposures we need to characterize, whether they are chemicals or particulates or air water pollutants, or whether they are social and social stressors. But what is that, and how do we go about making sure that there are studies of populations with these kinds of well-characterized exposures, and that samples have been collected and enough have been collected so they're available to then apply these new emerging technologies. It's not trivial. One can't just get this by going to the UK Biobank, having done a lot of work within the UK Biobank and with many cohorts and designed a number myself. There's a lot more that needs to be done than just seeing what would just go into samples that a hospital has or that a standard case control study has, and even that many prospective cohorts need. It requires another level of targeting, especially if you're going to start taking a half dozen different omics platforms. It could be very expensive. You really want to get the biggest bang for the buck. So who do you want to study? How are you going to characterize those exposures? Is the scientific community collecting the right kinds of populations, or enrolling the right kinds of populations with the right kinds of exposures? Are those exposures being characterized in the optimal way, and are samples being stored correctly? And I don't see any kind of a sort of uber approach to thinking about these questions and going through, well, here are the things that we need to think about. IARC can be helpful to a certain extent, because IARC has its class 1, it's 1A, it's 2A, 2B compounds, which are suspected carcinogens, probable or possible carcinogens, and a lot of attention could be put in trying to apply these technologies to populations with those kinds of exposures. I think that one of the things that might emerge from this, along the lines of an environmental atlas of sorts, is to try to be a bit proactive and think about, well, who do we need to study, and then try to go about doing that and coordinating it to some extent at least.
I think this is one of the challenges we see at NIH in the idea of like a coordination is that we have all of us initiating this study that's analogous to UK Biobank, but then we have all these disease cohorts and various institutes, and thinking about what role do these different populations play in understanding different diseases, but it's still very siloed in that sort of approach. So we addressed the sort of chemistry, the mass spectrometry part. I want to kind of turn it over to some of like the statistical and informatic piece. And Peter Kraft brought up this question about the sort of, and others did too, about the multiple comparisons. And, is our statistical framework appropriate for the type of discovery we need? When we think about when you're doing a multi-omic analysis, we tend to apply statistical genetics approaches to it, but now we're layering in, and you get to single cell, it gets very complicated. Is that I've always naively looked at like the idea of like network science as being the solution without knowing how it would work. But I'd like to hear comments of people about our sort of statistical framework, and are we missing things when we start bringing in these very complicated layers? I kind of think about like going to like the physics community and things about how you address this complexity. So I'd love to hear people's comments on this. You can jump in or raise your hand or something. Doug.
I think one thing that's often underappreciated in this as well is, there's this concern about the methods and whether the, theory the methods they're based upon is sound for the way we're applying them. But I think the other challenge we face with multiomics is that these are just massive data sets. And so when you think about all of the connections we're making among the different measurements, we can apply the best statistical methods we have and really try and interrogate the data. But where we often fail with a lot of our multi-omic studies, especially when we're incorporating unknowns measured in the exposome, is the actual interpretation of that. So we end up with these, massive hairballs and when we're doing network analysis, and how do we actually refine that into meaningful and new knowledge? Oftentimes we really end up just reverting back to the things we know and looking at the exposures we know and the metabolites and other omic measurements. But how can we improve kind of the interpretation science behind these multi-omic analysis as well? Because the data is just so complex, it's a huge challenge.
But your point about this, we tend to retract to our level of comfort. How do we stay uncomfortable? Because I think that's where we need to be for discovery. Ivana.
Yes, so I think this is actually a really good question. Even if we are thinking at the level of something like epigenome-wide association studies or transcriptome-wide wide association studies, so not GWAS, I think sometimes the statistical genetic methods are a little too stringent. And so then when you think about different layers, how do we think about reducing that stringency in a way that we are still adjusting for multiple comparisons, but not losing signal? And so I do think that, network analyses are a good approach. In single cell data, it's a little challenging because, you have more measurements, but a lot of the measurements are from the same individuals. So you really have to think about adjusting for the fact that you have repeated measures from each individual, and how do you adjust for that appropriately? So I don't have all the answers, but I do think as a community, we need to really think about how to appropriately adjust for multiple comparisons, but not over adjust.
Okay. Matt?
I think that even though one approach to the analysis of data will be a strictly agnostic approach, and that's the benefit of the omics approach. On the other hand, over time, if we can begin building up reference panels, that allows us to create some priors, and Bayesian or semi-Bayesian approaches may have utility in that we can define a priori certain processes or certain exposures if there's some a biological imprint of that exposure that we think are relatively high priors, and use that in the data interpretation. But at the end of the day, no matter what we do, and no matter what data reduction approaches we use or whatever priors we build into our analysis, it's trivial, but replication. I mean, that's critical, and that's what in the observational world, which is what we inhabit, that's what we're discussing. That's what's most important. And the thing is, along the way, not to get beguiled by false positives.
And I think this is like where, again, in the genomics community, because of the standardization, the idea of a replication validation set is pretty easy to do, because you can look at that data set and see that. And I wonder at times when we think about on the mass spectrometry side, is that would it be helpful to take a step back and just say, what if we prioritized 500 or 1,000 things to measure, and at least consistently measure the subset with a high degree of standard and harmonization? Would that be of greater value than trying to measure the hundreds of thousands with variability across all of it? And this idea, like what would kind of give us that sort of range? Dean.
If I can grumble at Nat for a minute for our statistical methods. So the beryllium toxicity in the lung is a very interesting one. And one of the individuals that was not in the figure that I showed on the cadmium was clearly an outlier. And we excluded the individual because the concentrations were so outrageously outside any distribution you could ever possibly imagine. And it turned out that this individual was a welder on space capsules. And so the exposures were so far out of line with everything else that if you followed any statistical procedure, you would conclude that that was not a human, that there was no possibility of having those levels, that there was completely outside the range of everything. And so this is an issue that when we're dealing with anything precision, we're talking about N of 1. And N of 1 in any sort of population study becomes a problem. And so that is something we just have to address in terms of our statistical approaches.
Understood. But the key is to have a larger N and have a wide distribution. Gary, I wanted to respond to the query that you just posed, which is really, I think, a really interesting one. I would strongly argue against an approach that truncates what it is that we're analyzing in the metabolomics world, assuming that we can get adequate precision for tens or now hundreds of thousands of compounds. I don't think we should ever be afraid of multiple comparisons. It's something that the frequentists make us all scared about. I have no fear of multiple comparisons. I just have learned through trial and error and having made mistakes along the way in believing my results too strongly back in the candidate gene days, as many other people did, without putting on the demands for replication. But I would never say, well, let's just look at a few things. Again, with the caveat that as long as we can measure things adequately, precisely, meaning the same degree of laboratory variation, I'm ignoring intra-individual variation, then the more the better. If we can, I remember a dinner more than a decade ago, and I asked, how many things are there that we can measure? And Dean said, well, probably a million compounds, and that was it's like the million molecule study. If that can be done and it can be done precisely, and the cost can be brought down, that's phenomenal. And very smart statisticians will figure out ways of analyzing that data. And fortunately, there are dozens and dozens of prospective studies in the world with millions of people enrolled, and they may not have repeat samples, and they may not have collected samples, not just in utero, preconception, even better, right? The ultimate cohort is a preconception cohort. Nevertheless, samples were collected at different times, speaking about secular time, in different places, different populations and the different ages, and that can be used even if you don't have repeat samples on everybody. So I would push the technology as hard as you can. The studies exist to be able to, for cancer, the studies exist. There will be enough cases to make this tractable. So the epi community has built this and are waiting for these methods to be further and further refined, and are very optimistic.
Okay. All right, so before I turn to Peter for the last question, I do want to point out that when we walk into our doctor's office, we are at N of 1. And so that is the ultimate goal, getting to that level of precision. So Peter.
Well, as it turns out, it's more of a comment than a question. So I just want to say the discussion we've been having around statistical analysis, I think I want to do +1 to all the comments so far. And I guess the way I frame it is it's, again, you have to think of what's the scientific goal, and that's going to drive your choices around how stringent do I want to be, do I want to be around adjusting for multiple comparisons. If it's like hypothesis generating, like I can follow this up easily in another study, or I can run a couple of assays in my lab, on 1,000 molecules, and that's like no sweat off my back. Then you can follow up 1,000 interesting leads from your observational study. If this is something that you would be taking to a clinical trial in humans, or you're proposing to use as a predictive score clinically, then you want to make sure that you haven't overfit. So again, it's like what analysis you want to run will depend on what's the scientific question. And then how you interpret that and how do you make sure that it's going to replicate and generalize is something that's worth thinking about, but it'll be context specific. And I'll just close by echoing the idea that from a lot of these studies, one of the key challenges is not going to be like, can we find associations? Can we generate fuzzballs? It's going to be making sense out of what those mean.
Well, Peter, I couldn't have asked for better closing comments for our panel discussion. So thank you very much. I'm going to thank all the panelists and speakers. This has been a very invigorating discussion. And we need to follow up on these ideas because there were many great ones. So thanks, everybody, and we'll see you after the break.
Great, thank you, everyone. We now have a short lunch break. We'll be starting precisely at 1:15 p.m. Thank you, Dr. Miller, and thank you to the panelists. That was a great discussion. We'll see you after the break.